Hello and welcome to our discussion of animal circulation. This is a glass tree frog. And glass tree frogs are aptly named since you can see their heart beating inside their chest. They remind me of Costa Rica when they were in the bathroom. Before smartphones, I would sit there in the bathroom in some of the field stations and watch these guys while I was, taking care of bathroom business. So I want you to think about this animal's heartbeat. What are going to be the evolutionary factors that might determine how fast this beat happens, as well as the size of its heart, or for that matter, any other animal? Well, there's a lot of things that can determine the size of an animal's heart or how fast it beats, but one thing that you probably cued in on was metabolism. There's a very distinct relationship between the heart rate of an animal and its metabolic rate, as well as the size of the animal, which we know is influenced by metabolism, and how large its heart has to be. So, physiologists recognize three different types of circulatory systems. First, there's really no circulation at all. So, something like a flatworm, a planaria, would take in food through its mouth and it would mix with any sort of fluids that are inside of the body. So, we call this a gastrovascular cavity. No distinct circulatory system yet. When you get to an insect, you now have a circulatory system complete with hearts that are moving fluid through a system of tubes, but these tubes are still open to the environment inside the body. So there's no clear distinction between what is hemolymph or blood versus any of the other fluids inside of the body. So we call this open circulatory system. By the time you get to an earthworm or a vertebrate like ourselves, you now have a system of pipes that completely separate blood from all of the other fluids inside of your body. So we call this the closed circulatory system. And again, you have a heart or multiple hearts that are moving blood in one direction through a series of pipes. So think about what's moving through this circulation. Well, of course, you're going to have respiratory gases, right? Oxygen, carbon dioxide. You're also moving nutrients through blood, right? Macronutrients, micronutrients, as well as other things like hormones that are signaling molecules moving through your bloodstream. Your immune system moves through your bloodstream. All of these things make blood essentially the superhighway, the interstate highway of your body. Now, how are these circulatory systems going to vary with metabolism? Well, you probably guessed it already. We talked about it already. Metabolism is going to drive for a more efficient circulatory system. So, bigger animals are going to have to worry about having a circulatory system that can meet every part of its body, even if it's such a large animal that those parts are far away from the heart, which is that central pump. Metabolism, the higher the metabolism, you're going to have more blood pressure, possibly to sustain that higher heartbeat, right? So all of these things are playing into shaping the circulatory system to meet the needs of the animal. So let's talk a little bit about open circulatory systems. Exoskeleton, that's the outside skeleton of an insect, like this grasshopper right here. That's waterproof, so it can retain all the fluids inside, right? And keep them from mixing with the outside. Now, there's no distinction between what we call blood and the interstitial fluid. So instead, we use the term hemolymph to describe this, the fluids that are in there. And there is a heart, there's also multiple hearts, and these are pumping fluid through channels, and they have these pores in the heart that are bringing fluid in, and then they're pumping them out through these tubes. But these tubes, these sinuses, they're they're just open to the inside environment. So they're not closed off completely. That draining will happen back through the pores into the heart. So there's circulation, but it's very much like the jets in a pool. It's where, water is being moved around, but it's not being moved around through a system of tubes. Now, do you think gases are being exchanged in this way? Well, you probably guessed already, but we talked about it already in respiratory system. In order to exchange oxygen and carbon dioxide, they have a system of tracheoles that are running throughout the entire body. So they're meeting their needs that way, in a way that's completely decoupled from the circulatory system. Right? So air is being brought in through those spiracles and moved around these tracheals. They don't need their hemolymph for any of it. However, earthworms and us, we have what you think of when you think of a circulatory system: a heart, some tubes that run around throughout your body, right? So this system of pipes. When you have a system of pipes like this, where the fluid is completely held within vessels, there's something you can do that you can't do if you have an open circulatory system. Well, hopefully you guessed it, blood pressure. Blood pressure is a thing now. You can control blood pressure, and we'll talk a little bit more about blood pressure, but this is what it's allowed vertebrates and things like octopi and squid, other cephalopods, as well as earthworms, I guess, to take over the whole planet. And we'll talk a little bit more about what that means. The other thing is to remember is, unlike the open circulatory system, you can now use this highway throughout your body for gas exchange. Now, you have a closed system, and as you have a closed system, what you see evolution doing is acting on the closed system, and the heart in particular, to eventually develop two separate circuits that are meeting different needs of the body. In a fish, for example, they have a two-chambered heart. One atria, where blood coming in from the body goes in there, it's deoxygenated, it goes into the ventricle right after, and then it gets pumped out to the gills. Gas exchange is happening, and then that oxygenated blood is making its way all throughout the body, which then is going to collect again and go back to the heart. Two-chambered heart, one loop. As you move up to an amphibian and a reptile, you're now seeing the beginnings of a three-chambered heart, where you're introducing a second atrium. And you're starting to separate between a systemic circuit that feeds the body and a pulmonary circuit that is in charge of getting that blood oxygenated again before being sent back out. So amphibians, they have a single ventricle, two atria. There's a lot of mixing of the blue and the red, shows up as purple here. That is the beginning of a pulmonary circuit. By the time you get to the reptiles, you have a much clearer division between your left and right. Your right being what's dealing with your deoxygenated blood, your left dealing with oxygenated blood. You're starting to separate out those two, less mixing. And by the time you get to a mammal or a bird, homeotherms, endotherms, quote unquote warm-blooded animals, you're going to start seeing a systemic circuit and a pulmonary circuit with a four-chambered heart. So this will ultimately be a very efficient thing for animals moving forward. So here is a diagram showing the pulmonary circuit and the systemic circuit, and a four-chambered heart. The numbers indicate blood vessels that are moving blood in and out of the heart. Blue indicates deoxygenated, red indicates oxygenated, 1, 2, 3, 4, right by the arrows that indicate the direction of blood flow. You know about arteries, you know about veins. Which of these numbers represent arteries and which of these numbers represent veins? Pick the best answer. Well, you probably know something about oxygenated versus deoxygenated, but that's not necessarily what you should be looking for. Those arrows are key. Arteries take blood away from the heart, and veins bring blood to the heart. Now, depending on whether you're dealing with the systemic or the pulmonary circuit, whether that blood's going to be oxygenated or not when it's arriving back to the heart will vary. So when you look at this, the arrows indicate that number 1 and number 4 are blood vessels that are leaving the heart. And when they leave the heart, that means they're an artery. Whereas 2 and 3 are bringing blood back to the heart, indicating that they're veins. So the correct answer was E. 1 and 4 arteries, 2 and 3 veins. Now, you probably learned about deoxygenated blood being veins and then arteries having oxygenated blood. And that is true when you're dealing with the systemic circulation. But when you're dealing with pulmonary circulation, that is reversed. And this is a thing that I've seen even on anatomy models and textbooks. They often get this thing wrong, or they forget to mention this, which is pretty terrible if you ask me. So animal hearts can vary. We already talked about the chambers of a heart, right? So fish have 2 chambers, birds have 4 chambers. For example, you could have 1 chamber like on the specialized heart of a spider, which is quite a weird looking tube. You could have 1 heart like we do, or you could have many hearts or auxiliary hearts, right? It really doesn't matter. They're all doing the same thing. And just like the human heart, the key thing that a heart is supposed to do is pump blood. In order to do this, the heart is basically a muscle that contracts. And as it contracts, it shoots blood out, fills back up, shoots blood out. And a heart is quite an amazing structure when you really think about it, and it's quite efficient. So when you look at a heart, you want to think about how blood is moving through it. So here are the 4 chambers of the heart. There's 2 atria, there's 2 ventricles, a right and left version of each. The right side of the heart is dealing with deoxygenated blood. Its job is to collect from the systemic circulation and send things out to the pulmonary circulation. And on the left side, you're dealing with oxygenated blood, getting blood back from pulmonary circulation and sending it out to systemic circulation. So they complement each other very nicely. So in terms of anatomy, we start with the right atrium because it's just a natural place to start. The superior vena cava is collecting blood, draining from the head and the arms. And the vena cava is the largest vein that you have. It's bringing blood back to the heart. Same thing with the inferior vena cava, which is draining your abdomen and your legs, etc. It's all coming back and feeding into the right atrium. As it's filling up, that blood starts to trickle through into the right ventricle through the tricuspid valve or atrioventricular valve, the right atrioventricular valve. And then when contraction of that heart happens, it's going to shoot blood through the right ventricle and through the pulmonary valve or the pulmonary semilunar valve into the pulmonary arteries that are then going to go right and left to the 2 sets of lungs, and then get oxygenated, coming right back through pulmonary veins that then drain into the left atrium. And the whole process basically starts, but on the other side. Blood's going to trickle through that left atrium, through the mitral valve or the bicuspid valve, and go into the left ventricle, and then upon contraction, it's going to shoot through the aortic semilunar valve this time, into the aorta, that grand artery that is then going to go and feed the rest of the body, right? And that aorta arches, the aortic arches kind of moves over the heart. It's a very distinctive structure. So that's the flow of blood through the heart. Now the thing to remember is a heart is a pump, and pumps, just like an electrical pump, work best when things are moving in one direction. So the last thing you want is backflow. So in order to prevent this, your heart is equipped with this network of valves. Now, it's important to remember that these valves are playing different roles depending on which stage the heart is in a contraction. So under normal circumstances, quote unquote relaxation, that time between the heartbeat, you have diastole. So the heart is relaxing, muscles aren't contracting per se. During this time, your valves are going to be either closed or open depending on which one you're looking at. And this is essentially going to reverse during contraction, or what we call systole. So during this time, we're talking about the ventricles contracting, forcing blood out of the heart. And the way this works is you have the 2 different types of valves. Between your atria and your ventricle, you have the atrioventricular valves, or the cuspid valves, the bicuspid on the left side, the tricuspid on the right side. During a contraction, the role of these valves is to essentially prevent backflow from the ventricles back into the atria. Last thing you want is things moving backwards. So during a contraction, papillary muscles on the inside wall of the heart are going to pull on these cords, and they're going to keep these flaps, which is all these valves really are, tight. So it prevents any blood from going back into the atrium. So imagine yourself trying to fly a kite in really windy weather, like a hurricane. You're holding on to the kite for dear life. You're having to inject force in order to keep that kite in one spot. That's what the papillary muscles are doing. This is all happening during diastole, right? So during diastole, the valves are open. But then during systole, when the contraction starts, those valves are going to be closed. And the force that's generated is then going to push through these semilunar valves, the pulmonary semilunar valve on the right side, the aortic semilunar valve on the left side. Those are the openings from the heart into the arteries. And those things are only open. They look like almost like peace symbols or Mercedes-Benz logos, and they're muscle. So in order to open them, you need to generate enough force to push back these flaps of muscle. So it's only during that contraction, that systole, that there's enough force to allow blood to force through and into the arteries, right? So this ingenious system is basically designed to keep blood moving in one direction through the heart, which is the way pumps work. So let's talk a little more about the details. This is a figure showing the outflow of blood out of the ventricle and during the course of a heartbeat. So during the course of a heartbeat, it's showing the flow. So this is a rate, a milliliters per second. You have the volume of blood inside of the ventricle. And then it shows the periods of time where the aortic valve and the atrioventricular valve and the left atrioventricular valve is open and closed. So this gives an indication of what are the events that's happening to blood during the course of a heartbeat. Of course, this is a cycle, and cycles happening over and over again need some kind of starting point. So the most natural place to start is during the initial phase of blood movement. So the atrium contracts a little before the ventricle, and we'll talk about that in a bit. So during this atrial systole, you're basically forcing blood to leave the atrium and trickle into the ventricle. So if you look at the volume, it's increasing during this time, during this number 1, right? But you don't have any outflow yet because nothing's leaving the heart yet. So during this time, the atrioventricular valve has to be open because blood is trickling through into the ventricle. The aortic valve is closed. But then what happens is enough blood gets in to where the atrioventricular valve closes. So you have this period of time, this little small window of time, where both the AV valve and the aortic semilunar valve are both closed. During this time, the volume present inside of the ventricle remains the same. So isovolumetric contraction point is right here. This is during a systole event. So the force is starting to generate, but not enough to cause anything yet. So you have this period of time where both valves are closed, isovolumetric contraction point. Then what's going to happen is enough force is going to be generated that it's going to essentially increase the intraventricular pressure, and then it's going to cause the blood to push through the aortic valve. And when it does this, that valve obviously opens. You can see in the chart that the ventricular outflow has this big peak, right, where you're suddenly flowing a lot of blood out of the heart, and then it goes back down. At the same time, that volume inside the ventricle is very high and starts to go back down. Well, once this happens, your AV valve, with the blood leaving the heart, there's going to be no more force on the AV valve, so it's going to close right back up. But the aortic valve, the AV valve has not yet opened up yet. So again, another period of time where both valves are going to be closed. So both the AV valve and the aortic semilunar valve are going to be closed. And then this time, the heart is starting to relax. So we call this the isovolumetric relaxation point. So there's 2 periods of times during the course of a heartbeat where both valves are closed. The isovolumetric contraction, isovolumetric relaxation point. When the valves are closed, there's no changes in volume, there's no changes in flow. What's going to happen next? Well, what's going to happen next is that blood's trickling into the atrium, right? And it's going to cause the atria to open that AV valve again. And now that blood's going to start flowing back into the ventricle, and you're starting to see that increase in ventricular volume, right? Nothing's leaving the heart yet, so that's still static. So you're starting to set up the next heartbeat at this point, right? So it's important to remember the relative events of blood moving through the heart and when they occur. So what's controlling this heartbeat? Well, it depends what kind of animal you are. If you're something like a crayfish, a lobster, a spider, or a horseshoe crab, or a scorpion, or something like that, then the nervous system is controlling your heart in what we call a neurogenic heart, right? And basically it's saying that your heart is going to be innervated by neurons, 9 neurons, that form what they call a cardiac ganglion, which is basically just a bunch of neurons and cell bodies that are working towards a single purpose. So the beat is determined by the central nervous system, which is informing all of this. So either the brain has what they call a central pattern generator, which we'll talk about in the future. And that central pattern generator is doing exactly that. It's generating a pattern using your central nervous system. And it's going to basically determine when the heart is beating. In the case of vertebrates like ourselves, the heart has decided to take on the job itself. So it's decoupled the heartbeat from the nervous system, not quite entirely, but almost. And it does so because there's specialized muscle cells that are what we call autorhythmic, meaning that they can generate their own pattern. And they do this by essentially undergoing these spontaneous depolarization events. And these generate action potentials. So action potentials, we'll talk about them in more detail in the future, but you probably already know every time a neuron fires, what that means is it's generating an action potential. So as it generates an action potential, it's depolarizing, meaning that it's changing the charges across the membranes of that cell. This specifically is happening in the sinoatrial node, which depolarizes a ton of times in a minute, 100 times or so. The sinoatrial node is the SA node, and it's more popularly known as the pacemaker. These are the pacemaker cells. They're located in the right atrium, and what they're going to do is they're going to send a signal that's going to move through the right atrium and into the left atrium, and eventually converge on this place known as the atrioventricular node, the AV node. That signal is then going to get sent into the ventricle through a network of bundles that terminate in essentially the papillary muscles, and as they move through in a wave, they're causing contraction to happen. So an important thing to remember is that 100 times a minute is actually a pretty high heart rate for a human being. So that would be your resting heart rate, except that your nervous system is still involved. What the nervous system does is it puts the brakes on your heart rate and will release the brake if it needs to. So it's still being modified by the sympathetic nervous system and the parasympathetic nervous system. So the sympathetic nervous system is more your stress part of your nervous system. Parasympathetic is more your normal sort of day-to-day unstressed, if that's even a thing, parasympathetic nervous system. So when you're under normal circumstances, your parasympathetic nervous system is putting the brakes. When you're stressed and you need to increase your heart rate or do an exercise or something, your sympathetic nervous system is lifting that brake. That's what happens. But your heartbeat is ultimately determined by these autorhythmic cells. So how do these autorhythmic cells do it? To generate an action potential, all you're really doing is changing the charge from one side of a membrane to the other. I tell you before, things that happen in your life, it's either because something changed its shape or something changed its charge across a membrane. So that's what these guys are doing too. Now, when we talk about charge and electricity in physiology terms, we're almost always talking about ions. And we're talking about changes. All we're talking about is channels opening and closing, permitting ions to move in and out of a cell. In the case of these autorhythmic cells, they generate what's called a pacemaker potential, which is kind of like an action potential, but involves a slightly different mechanism. We'll talk about action potentials in a future lecture, But for pacemaker potentials, the reason they can do this in a spontaneous sort of way is because they have these sodium channels that are funny. And all that means is they're funny because they've got some weird molecular clock that causes them to open at random, essentially. When these funny sodium channels open up, you get this sudden influx of sodium into the pacemaker cell. This influx of sodium is driven by the fact that there's a very large gradient between the outside and the inside of the cell. So when these channels open, that sodium is moving through this gradient very quickly, making the cell depolarize or become more positive. So that's what you see here. You see the membrane potential. The resting state is probably around minus 70 millivolts. And then as these sodium channels open up, they start to become more positive or less negative. That's a byproduct of how we measure it. We'll tell you about it later. Well, this sudden change in charge then causes another set of channels to open up, calcium channels. And these T-type calcium channels open up and they stay open a lot longer. So they're getting a lot more calcium to come in, which causes that full-on action potential change, causing that rapid depolarization. Well, that can't go on forever. The cell wants to maintain integrity. So what does it do? It closes those channels down and then opens up these potassium channels because potassium is much more present inside of a cell than outside. So as it opens up these channels, that potassium is starting to leak out to its own gradient, making the cell less positive because it's getting rid of positively charged potassium. And as a result of being less positive, another way to look at it is becoming more negative. So it's basically coming back down. So that is the course of a pacemaker potential, and the whole process is going to start right over again. And this happens 100 times or so a minute. So what about muscle cells? Myocardium. So the cool thing about cardiac muscle is the way it's organized. It contracts, and the goal is not necessarily to move things in a direction like you have with skeletal muscles, but to induce a complete squeeze on the heart to get rid of as much blood as possible. Now, the way it can send this signal quickly is by having muscle fibers or muscle cells joined to each other, but are able to communicate with each other through gap junctions. Do you recall what gap junctions are and what they're doing? Okay, so muscle cells are very large cells, and where they meet, it's called an intercalated disc. They're joined together by desmosomes that allow them to kind of stick together, but they have these gap junctions. And if you recall from probably general biology, gap junctions are basically channels between two cells. We already talked about potentials being caused by ions. These positive ions are now going to be inside of the cell. Those sodium and calcium ions are going to be able to move through these gap junctions and start to trigger depolarizations as they move through cell to cell to cell. Which is why whenever a contraction happens, it's seen as a wave. So as one cell produces it, it basically sends that signal to the next cell and the next cell and the next cell. That's what gap junctions are for. They facilitate that. The end result, this wave-like pattern of contraction where all of these cells are contracting together and working together to squeeze as much blood out of the heart as possible. So it's a really cool thing about myocardium, cardiac muscle. Now, if you look at cardiac muscle on the left and right side, you'll notice that there's a big difference in terms of how thick they are. So if you were to do a cross-section of the ventricles, the right side is a lot thinner than the left side. There's a reason for this. Here are 3 choices. Well, it has to do with what they're doing. The right ventricle is in charge of sending blood to the lungs as part of the pulmonary circuit. The left ventricle sends blood to the rest of the body. If you think about it, the lungs are right by the heart. So it makes sense that that pulmonary circuit wouldn't need that much force to send blood out there. Whereas the left ventricle is feeding the entire body through the systemic circulatory circuit. So it needs be able to generate a much stronger force to be able to send blood through the arteries there. So the differences between these two is B, relative differences in the lengths of the pulmonary and systemic circulatory circuits. Okay, so what happens during a cardiac cycle? So a cardiac cycle defines an entire period of contraction. So we talked already about the blood moving through. Now we're going to talk about the electrical events that are happening and how they get recorded in an ECG or an EKG, an electrocardiogram. The K comes from the fact that this is an old German technique and they spell cardio with a K. It sounds cooler too, EKG versus ECG. As an abbreviation, I think EKG sounds cooler. And this is what produces that blip that you see in the movies when the person's in the hospital and you hear beep, beep, beep. That's the EKG. So we'll start with the sinoatrial node. Autorhythmic cells start to depolarize. Those ions that are flooding into the cells start to move through gap junctions and eventually will take over the entire right and left atrium. So during this yellow coloration in this diagram, I want you to envision that there's contractions happening where it's yellow. It's eventually going to contract the right and left atrium together, forcing blood into the ventricle. Well, the signal is eventually going to converge at the AV node. And during this time, that signal is now going to be sent through the ventricle. As it's sent down the bundle of His and down the bundle branches and into the interior, during this time, the atrium is starting to relax. Now, as this yellow is happening in the ventricle, that contraction is also happening. And then eventually, that's going to go away. So when you follow the events and look at it on EKG, that first atrial excitation, well, that's electrical events that are going to be recorded as a little blip called the P wave. Now, after atrial excitation, after that contraction happens, you get the beginning of a much sharper peak associated with the depolarization of the ventricles and the contraction of the ventricles. And this is known as the QRS complex, which stands for the 3 points on the triangle, with R being the high point on that peak. Then, as the signal goes away and the ventricles start to relax, you have a third wave that comes up, which registers as what's known as a T wave. So that's the little blip at the end of the ventricle relaxation. Important thing to note, EKGs measure electricity and changes. So the P wave is measuring atrial depolarization. The QRS complex is measuring ventricle depolarization. And the T wave is measuring ventricular repolarization. So here's the question. Where is the wave for the atrial repolarization? This is one of those trick questions. You know me. It's none of the above. What? How come there's no atrial repolarization wave? Well, the P wave is the atrial depolarization. The QRS complex, or waves, are the ventricular depolarization, and the T wave is the ventricular repolarization. Well, remember what I said. The atrial depolarization, the relaxing of the atrium, is happening at the same time as the ventricles contracting. Ventricles are bigger and are going to generate a much bigger signal. So the QRS complex is actually masking the atrial repolarization wave that you would otherwise see. Now, in case you're curious about the U wave, not everybody has this, but sometimes there's a delay in the valves closing, and there's electrical activity associated with that sometimes occur as a U wave. That happens at the end. But it's not something that everybody has, and it's not diagnostic of anything in particular. Just thought I would mention that. Okay, so we talked about the natural pacemaker, the sinoatrial node. Well, what about an artificial pacemaker, which is basically designed to have two leads that cause electrical signal to run from one to the other. Those things are usually implanted in your upper chest, and then those leads are run through your subclavian vein and into your superior vena cava, which then go into your right atrium. What kind of wave are they supposed to generate? What's their job? Well, hopefully you guessed it. They're a pacemaker, so they're acting like the sinoatrial node. So their job is to generate the P wave, with the assumption being that once you generate that P wave, the rest of the events will follow. And that's how they work. So now every time your heart beats, it's doing so in response to the rest of the circulatory system. Your heart rate is beats per minute. But we're not talking about just the number of beats in a minute. What's also important is the volume. Every time your heart contracts, it releases a certain amount of volume known as a stroke volume. One heart stroke produces a set amount of blood moving through your circulatory system. Now, there's an interesting relationship between how much blood is present in your body, i.e. your blood volume, which is far from a constant thing. You know this. If you're dehydrated versus, very hydrated, you're going to have different amounts of blood volume. But your heart is still one organ having to do the same job over and over again. So it has to have a way to adjust what it's doing in response to this. So we talk about the Starling Law of the heart. And that is the idea that you're going to match your cardiac output with your blood volume. So cardiac output is the product of your stroke volume and your heart rate. So what is going to be the units for cardiac output? Well, you probably guessed your heart rate is beats per minute, and your stroke volume is the volume of blood in a single beat. So by multiplying the two, cardiac output is going to be your volume per minute, usually milliliters per minute. So your heart muscle, your myocardium, is actually quite elastic. And whenever blood enters in there, it imposes a pressure on the inside walls of your heart. It's causing a stretch. And that stretch produces a force. Being elastic as it is, when your heart stretches, it induces a force of contractility. So the more volume, the more it's going to stretch, the greater the contractility. Then during the heartbeat, when your heart actually starts to contract, that contractility is going to add extra force to that heart rate, which is going to then change the amount of blood leaving your heart and changing your cardiac output. So this degree of stretch we call preload. So preload is essentially the amount of contraction the amount of stretch that your heart is going to experience right before contraction. So if you look at this graph right here, it's showing the left ventricle volume and left ventricle pressure. And basically, the greater the amount of blood volume in the ventricle, the greater the amount of pressure it generates. It has higher preload. And the reverse is true as well. Low amounts of preload, in other words, low blood volume, you're going to get less force. So this is an excellent way of matching your heart's pumping action with the conditions of the rest of your circulatory system to match homeostasis. Next week, we'll talk about water balance and how that plays into blood volume. But this is an important concept that needs to be understood. So I'm starting to feel my age. And, I've done I've done quite a bit of skydiving. I've gone bungee jumping a couple of times. I love roller coasters. But lately, anytime I lift my head really fast, if I'm down on the ground, say doing some landscaping or something like that, I'm going to get a little lightheaded, and I hate it. And then I think about giraffes, which are one of my favorite African animals, and how they can drink and then suddenly just lift their heads when they're done and walk around like nothing's ever happened. What is it that giraffes have that prevent them from getting this vertigo that I end up getting? So take a second to think about that. Well, I'm sure you've picked up on the main thing, which is differences in blood pressure. Obviously, giraffes have a larger heart than me. They also have a longer neck than me. So they have to rely not only on this larger heart of theirs, but they also have to have a way to manipulate blood pressure to prevent them feeling lightheaded. The lightheadedness comes from the fact that the blood that has been pooling in your head is suddenly drained away too quickly, and it leads to this feeling of dizziness that we call vertigo. Giraffes don't feel vertigo because they don't allow blood to pool in their head, and when they do raise their head back up, they drain it slowly and not quickly like I do. So the way they do this is twofold. One, through maintaining very high blood pressure at all times so that they're constantly moving stuff through even when they're down low, like that, their head, and also by a network of valves that prevent blood from draining too quickly. So there's a relationship between height and pressure. Blood pressure is going to be determined by your heart pressure. And the further away you are from your heart, the less that pressure is going to be felt. When you're talking about pressure down at your feet, that's where pressure is the highest, because not only is your blood pumping that way, but you also have the added effect of all of the volume of blood that's above it. So if you think about a tube, right, at the top of the tube, blood or water is going to have a certain pressure. But as you sample pressure further down, you're not only dealing with that same pressure as on the surface, but also the added pressure of gravity and all of that weight of the water right above it. It's why when you go scuba diving, the deeper you go, the more pressure you feel, right, in water, because you have the weight of the whole water column above you. So depending on where you are in your body, you have to deal with different types of pressures simply because of its position relative to your heart. So this is a fundamental thing that have shaped how our blood vessels operate. So there's three types of blood vessels. There's arteries, veins, and capillaries. Which one of these three are responsible for the diffusion of things in and out of our circulation? Well, you probably guessed it's the capillaries, right? So anytime we talk about things moving in and out of blood, we're talking about things happening at the capillary level. Your arteries and your veins, on the other hand, are simply pipes, transport. They could be really large or they could be smaller. Small arteries are called arterioles, small veins are called venules, kind of the same thing. There is a difference between these types of arteries and veins. Arteries are very thick. If you look at a cross-section of them, they hold their shape very well. And that's because of the middle layer that they're composed of. They're composed of three layers: an outer layer, an internal layer, and a middle layer, which is made of smooth muscle. They have a very thick middle layer, which allows it to retain its shape. It also allows contractions to happen that can control blood pressure, vasoconstriction versus vasodilation. Veins, on the other hand, have a much thinner layer, so when you look at them, they tend to collapse on themselves, right? So even in a cross-section here, you see a very big difference between the arteries and the veins, right? Veins don't maintain that circular shape very well because that middle smooth muscle layer is a lot thinner. One thing veins do have, however, that arteries don't have, are valves. And these valves are basically there to prevent backflow. We'll talk a little bit more about them when we address this next question. What's driving blood pressure in the arteries, and what's driving blood pressure in the veins? Well, in the arteries, it's pretty straightforward. It's the heart. Whenever the heart contracts, it's generating the pressure that is felt in the arteries. So blood pressure in the arteries is going to be determined by that, as well as by the vasoconstriction and vasodilation, which we'll talk about later on. Whereas in the veins, it's something else entirely. If you recall, heart contracts, sends blood through the arteries, there's pressure, but when you get to the capillaries, these small vessels, there really isn't much pressure there. And when they collect and they reform veins, whatever the blood pressure that came from the heart is now gone. The pressure for veins is actually generated by skeletal muscles. So if you think about your feet, whenever they have blood down there and you're bringing it back to your heart, the way that happens is your veins are moving in and out of your skeletal muscles. And the muscles in your leg are constantly contracting. And as they contract, they push a little bit of blood up the veins. And that blood does not fall back because of those valves I mentioned earlier. So that's what's driving your blood pressure in your veins. It's actually your skeletal muscles, not really your heart at all. So if you're watching this video, you probably don't have access to a group. But I want you to take a look at this figure showing blood reservoirs, if you will, and then a bunch of graduated cylinders that are each being fed by different tubes and the amount of blood inside them. You could pretend that each of these graduated cylinders is, say, a different organ or a different cell type. I want you to think about what is it that's ultimately determining what each organ or cell is going to get. And I'll give you a hint. That's a Guy-Fawkes mass. Okay, well, no matter where you are around the world, whenever there's some kind of riot or something, you always see these masks come out. And they're named for an Englishman who engaged in a little resistance against the Queen of England, and then, got what was coming to them from the Queen of England as a result. Not the Queen, Elizabeth, an earlier one, Victoria. But Guy Fawkes basically led a resistance movement. And resistance is key here. Resistance is the obstruction to flow. Blood wants to flow. And how fast it flows, its flow rate, is going to be determined by the differences in pressure on either side. It's going to be determined by the radius of the blood vessel it's moving through, as well as how thick the fluid is and how far it has to travel, or what the rate is. All of these factors together will generate a resistance to flow. The other thing to remember is the relationship between radius and flow can be explained by understanding laminar flow. And laminar flow is basically movement through a tube. So if you look at the flow of a fluid through this schematic here, the further you are from the edges of the tube, the less resistance you encounter. So the faster you flow. So that's what laminar flow is describing this dynamic. So that's why by vasodilation, you're minimizing the resistance to your flow, right? By engaging in vasodilation, making the radius bigger, you're allowing more flow to happen. By making it smaller, you're decreasing the flow, increasing the resistance, and increasing the pressure inside of that tube. So ultimately, artery or vein, you're going to end up in a capillary. And it's really the dynamics of the capillaries that interest us in terms of physiology, because that's where everything takes place. So here's a capillary, and the capillary is going to be subject to a variety of different forces and pressures. So whenever we talk about fluid leaving a capillary, which it usually does when it's close to the arteries, because the arteries are bringing things that are needed by the body, that fluid leaves the capillaries, we talk about the process of filtering. Whenever fluid is entering the capillaries, as it does towards the vein side of things, right, then we talk about that being absorption. What determines it are essentially 4 different processes. The first type of process are hydrostatic processes. There's 2 opposing forces here. There's the fluid inside of the capillary, which is inducing a capillary hydrostatic pressure, which is the pressure that fluid wants to get out of the tube. And that's being opposed by the interstitial fluid hydrostatic pressure, which is the fluid forcing itself onto the capillary. So they're opposing each other. At the same time, you also have osmotic forces that are determined by the concentration of solutes in the plasma portion of the blood, right, in the fluid portion, as well as in the interstitial fluid. So the hydrostatic pressures are abbreviated as P. The osmotic pressures are abbreviated as pi, right, the math symbol, the Greek symbol. The osmotic pressures in the blood are being driven primarily by protein concentrations of albumin, which is one of the most common proteins in your body. And basically, the more concentration, the more solute you have, the more protein you have, the more concentrated it is, the more you're drawing water into the tube, right? Or out of the tube, depending on where the osmotic force is higher. So the outcome of these 4 is going to produce your net filtration pressure. So what are the pressures driving what are the forces driving your fluid out of your capillary? Subtract the forces that are driving the fluid into your capillary. If your pressure to leave is greater than the pressure to stay, you're filtering. If it's the reverse, it's absorption. You can actually calculate this. So here's a set of conditions. PC equals 15 millimeters mercury, assuming that pi C is 11, assuming pi IF, interstitial fluid, is 3, and hydrostatic pressure in the interstitial fluid, PIF, is 12. What is the net filtration pressure? And if you get this value, you should be able to tell if it's an artery or a vein. But here's the thing. I'm not going to sit here on this video and wait for you to do a math equation. So do it, try it on yourself, let me know when you find, and let me know what your answer is, and we can talk about it. So come find me. In the meantime, let's move on. Now, osmotic pressures don't tend to vary much. Albumin's pretty constant in the blood, but hydrostatic pressure does vary. So, and remember, a capillary is basically joining an artery with a vein, and it's the process in between. On the closer to the artery side, fluid is forced out of the capillary because hydrostatic pressure is higher. But as you reach a certain point, that hydrostatic pressure starts to decrease. This is the pressure inside of the capillary, and as a result of that, the pressure to move fluid into the capillary increases, causing reabsorption to happen. So as you proceed from an artery to a vein, your capillary is doing very different things. And this is important because your blood vessels are not static. They change through time. So when you want to change circulation, you can vasodilate, you can vasoconstrict to change resistance, and that's a quick short-term thing. In the long term, however, if you need to change your circulation, you might change the blood volume through changes in your water balance, which we'll talk about in a future video. Or you can change the number of capillaries that are being involved. So capillaries are usually in the form of capillary beds. They're small vessels. They move one red blood cell at a time, as these images show. And depending on the situation, if you need to reabsorb more, you might open up your capillaries by opening up these sphincters and allowing the blood to flow through all of them. But if you want to restrict blood flow, you can do that by closing those sphincters, thereby only sending blood through a single central capillary. Really depends. A lot of variety. So just to revisit what we asked in the beginning about evolutionary factors that determine the size of hearts and how fast they beat. Remember, hearts are dynamic. So evolutionary factors are always going to favor things that increase the efficiency of the pump. They're going to impact blood pressure and resistance. And these are going to change um, ultimately how the heart beats, how the cardiac output changes through time, how the heart rate changes. So all of these are there to make sure that your heart and your circulatory system are functioning to the best capacity possible. And with that, I will see you in the next video.